is entitled MALDI, From Birth to ... I don't know if I had the right number of dots there or not. And it'll be presented by Franz Hillenkamp of the University of Münster in Germany. Franz?
Thank you. Well, certain experiences that Peter was presenting, we had also, so there is some repetition in there. And where do I... Oh.
Which one were you?
14.
This one?
I think so, yeah. Try that. Yeah. So. Okay, so that's a ... When I was thinking up the title, actually I thought Marvin had provided a title and I would adjust my talk to his title, but then it turned out he didn't. And when I finally sat down to compile the slides, I thought it was from birth to, certainly not to death yet. I think MALDI is still a very viable technique. Actually, Peter was coming in from the protein side. We were coming in from quite a different angle originally. It was in the late 60s that I was at a Mardi Gras party in Munich, and I saw a guy sitting in the corner and he looked bored. And I was somewhat bored. It was not a good party for a change. And so I sat down with him, and over a couple of beers, we exchanged information about who we were and what we were doing. It turned out he was a physiologist from Freiburg University, and he was very interested in the distribution of calcium ions in heart muscle cells. So what he needed was a technique that had a spatial resolution way below the size of a cell, that is a micron or better, and some sensitivity, analytical sensitivity. And so the next day he came to my laboratory. I knew lasers would be good for that, and I was a laser guy actually, because I knew how to focus a laser down to less than a micron in the UV. We discussed then, if we had the laser and the spatial resolution, what would be the best way to analyze what's coming off the sample. And we first thought about emission spectroscopy, but then looking at some of the literature, it appeared to us that most probably the sensitivity wouldn't be good enough. And sensitivity is a constant topic throughout my talk, actually. And so mass spectrometry, none of us knew anything about mass spectrometry, so it looked promising. And we set up a laboratory setup that you see here, and you see we were working on thin sections as they are used in electron microscopy, and the sections were mounted in about this plane here, on an electron microscopic grid underneath a quartz window which acted as the vacuum window. And in this case, you already see the time of flight tube down here. We started out actually with quadrupoles, but when we found out that it took us more than a day and consume at least a dozen samples before we had accumulated a spectrum, we gave up on that. And I don't remember how we rediscovered time-of-flight mass spectrometry, but somehow we did, and it looked, from the very first moment, quite attractive. So back here is the laser, which was at that time a frequency doubled ruby laser at 347 nanometers in the UV. And this is a microscope that we assembled from pieces we had from microscope taken apart to look at the sample. And so that was our very first setup, and we did get relatively decent spectra. But the question was, my then boss didn't like that work. So he said, well, if you can get money for it, you can do it, but I'm not going to give you any money for it. And so we wrote a proposal to VW Foundation, and it's in German, and I didn't want to translate it because this is the original. later, when we showed the feasibility of what we wanted to do, we went back to the program officer at VW Foundation and asked him why in hell had he been funding our proposal. And he just laughed, and he showed us one of the reviews, and it says this project is at the limit of the physically possible. The expected sensitivity has never been reached so far, and in all likelihood will also not be reached by this project. So that was quite negative, and we asked him, why the hell did you give us the money? And he said, you know, I had been looking at proposals all week long, and they were good proposals, but they were not really exciting in the sense that it was something absolutely new. And it was Friday afternoon, and I was finally looking for something that we would now call high-risk, high-yield project. And we just were lucky that our proposal was on his table, and he gave us the money, though he wanted to give us a little bit of a caveat, but also cover his back. And it says, with the acceptance of these funds, you take on the risk or the responsibility for the high risk of a successful completion of the project. So, I mean, how we could do that, I don't know, but that was what he wanted to do. So the question is, how about this review here? Was the reviewer just not able to understand what we were up to? No, we were naive and young at that time, and we were stupid enough to include in the proposal an explanation of how we thought it would work. And the explanation was that we knew with a laser, if you focus it down, you can bring it to optical breakdown. So you can generate a plasma. And we said we'll generate a plasma and extract the ions from the plasma. It didn't take us long to find out that if you do that, you get no real spectrum. You get ions all over the place. You can't extract them and what have you. So you have to just tickle your sample. And that's one of the secrets, I think, of MALDI if you want to do it right. So I was never happy with the expression desorption, because desorption is actually something that's adsorbed at the surface. So it is a volume process, but I may come back to that later a little bit. So one of the things we did, then we looked at tissue sections, and this is a kidney section, and these are spectra to look at the different sodium and potassium concentration in these different areas of the kidney. I don't want to go into it. I just wanted to point out, we'll hear more about MALDI imaging later today. This was, we called it LAMA, Laser Microprobe Mass Analyzer. It was LAMA imaging, and we could do it with this high resolution because we were only looking at elemental ions and not molecular ions, and that makes life quite a bit easier. You also see that the spectra were on strip chart recorder. Originally, I would go into the laboratory, pull all the shades down, sit there for 10 minutes or 20 minutes to completely dark adapt, and then I would stare at this small screen of the then fastest oscilloscope and saw this the spectrum pass by. And when I thought we had some area on the sample that was interesting enough, we would take the then fastest Polaroid films to take pictures of that. When we took these, we had already the very first digital converter, analog to digital converter, the Biomation 8500 or so. So at least we could capture a small part of the spectrum electronically and then read it out on a strip chart recorder, and then we would have these strip charts on the floor and use a meter stick for mass calibration. So it wasn't very fancy, but I'm standing up here to say that if it would not have been for the tremendous development of electronics since 1970 or so and now. None of these techniques would ever have made it to a really useful analytical technique. So developments like that rarely stay completely by themselves. There are other developments that have to come along with it, and the electronics, I think, was extremely important. Also, some years later, we started a collaboration with a company. Actually, the Federal Department of Science and Technology forced us. They said, if you don't have a partner, industrial partner, we're not going to fund your work. And so we struck a deal with Leibniz in Cologne, which at that time was making XPS and similar analytical instrumentation. And they were willing to build a Lama instrument. And this is their Lama 1000. It already looks pretty professional. Actually, at that time, we had already a top illumination and not a transmission illumination, because it was mostly used to look at wafers, large wafers, to see at contaminations after they had been structured. And again, you can see that here, of course. This was now a horizontal time-of-flight tube, and I think it was the first TOF, Peter, I have to correct you, I think it was the first commercial TOF after the Bendix had originally abandoned their TOF instrument. And it certainly was the first TOF with an ion reflector that had been invented by Mamure and published a few years before. And, well, I was sold, I don't know, 50 or so of these instruments. So, to change topic a little bit for a moment, I think, and that's, I think, true for Peter as well and many others in those days, I was extremely lucky that many of, or quite a number of people that started out as collaborators on certain aspects of our projects eventually turned into personal friends. And this person who came up with the idea of the calcium measurement was actually Raimund Kaufmann, and I would like to, he died of a brain tumor a couple of years ago, unfortunately, and I want to give him credit of being strongly involved in the early days. At one day he was just banging at erythrocytes, red blood cells, and he saw a signal at 56, and he was jumping up and down saying, I'm seeing the iron there. But then we told him that the isotopic pattern was in no way the isotopic pattern of iron. So then we kept teasing him about the organic iron that he could see in the mass spectrometer. So at that time, he was moving on to Düsseldorf, and I was moving on to Frankfurt, and collaboration got a little more difficult. And we decided that we would sort of split the work. He would pursue the biological applications, and I would do something else, because as I said, sensitivity was an important factor. And when we carefully looked at our spectra, we realized that the limitation for sensitivity was not that we didn't generate enough ions. It was the grass on the baseline of the spectra, what we now call chemical noise. So there was a peak on every mass, and once the signals got lost in the chemical noise, we couldn't see them anymore. And we realized that this chemical noise had some long wave modulation on it. And when we began to think about it, the only reasonable explanation was that these were fragment ions from the epoxy resin that's used to embed the tissue sections in. This was the first time in my life that I ever heard of and thought about molecular ions. I was coming from physics, and molecular ion was not something that I had been told about. And so to verify that our concept was right, we were just using a different epoxy, and sure enough, this modulation changed in a systematic way. So I realized there are organic ions, and it may be worth to pursue them. And what do you do? The first thing, in order to be reasonably systematic as a scientist, I decided to look at all the, I think, 26 or so common amino acids. And we took spectra of all these amino acids, and we tried to publish that. And the paper never appeared because I think after the third attempt to get a journal to publish it, it was turned down. We left it there. The next slide will show. Now, we were bold to go on to dipeptides. And what we found was that to get spectra, this E0 is the laser energy, or actually fluence, in the laser spot. And we found, and we were at that time at 266 nanometers, the quadrupled wavelengths of the, or quadrupled frequency of the medium YAG laser. And for tryptophan, we had the lowest energy we needed. And the more we got into the aliphatic ones, the higher the energy we needed, and the more fragmentation we got. In this point, it helped me a lot that parallel to the mass spectrometry, I was doing laser work in clinical medicine, and I had been interested in the interaction of lasers with tissue, and I knew something about the absorption, so I knew that the aromatic residues of tryptophan and tyrosine are absorbing at 266 nanometer, but the aliphatics don't. So the result was not that much of a surprise, actually. And so that was the status of knowledge at that point. And in 83, the next, well, I should first say about my work in laser medicine. I did most of that at the Wellman then Laboratories of Photomedicine in Boston, and the then director and head of the Department of Dermatology at Mass General and Harvard Medical School, John Parrish, is another one of my collaborators that turned into very good personal friends. But in 83, there was a real hit, and that was Michael Karras here. By the way, he would have loved to come, but he's organizing this year's annual meeting of the German Mass Spectrometry Society, which is taking place right this week in Frankfurt, so he couldn't make it. This was taken later at some award ceremony. I don't know what we are laughing at, but something, I thought it was a good picture of certainly him. And I hired him as a postdoc because I thought I understood the lasers, I understood the photophysics, but I didn't know much about chemistry. So I needed someone in chemistry, and he had just gotten his PhD from Bonn, from Beckay actually, who Peter mentioned. And so from day one on, we just got along with each other extremely well. We kept debating things. And many people asked how can they get along because they're always shouting at each other. But it was scientific discussion. It was never anything personal. And we didn't give up or finish until we thought we had a solution that satisfied both of us. So Michael is certainly very important in the development of MALDI, and is now, of course, as I said, a personal friend. So about a year later, he came one day to my office and showed me the spectrum. And he said, I have something interesting for you to look at. And I said, well, what is it? Well, he said, it's a mixture of alanine and tryptophan, not a dipeptide, but it was a mixture because in the dipeptide spectra, we had seen some odd features in which we could not understand. And when we talked to a biochemist, he said, oh, well, if you buy a dipeptide from a company, you wouldn't believe that it's pure. There are lots of amino acids still in there. So to follow up on that, we did a systematic dilution series, and this was part of that. And I said, yeah, that's okay. But he said, you know what? I took the spectra at the laser fluence that we need for tryptophan. but here you see the alanine is there as well. So it took us about 5 minutes to realize that something strange had been going on, that the alanine, which should not have appeared at this laser fluence, was riding along piggyback with tryptophan. And for a while, we tried tryptophan with other substances, sugars, for example, and it turned out that this was more of a general principle and was worth pursuing, which we then did. This was the birth of what we now call MALDI. Let me also show you something. Peter mentioned Frank Fields, who was one of the giants in mass spectrometry in those days. And even in '86, he still published something saying, my personal feeling is that to do so may well require the discovery of some new technique. Now, ESI as well as MALDI came, so to speak, out of the woods. Nobody would have predicted either of these techniques to be very useful in mass spectrometry, and it was just a combination of systematic investigation and luck and serendipity that we could do these spectra. So, in the end, and I don't want to go into any details, in the end it turned out in most cases you're better off if the matrix is there in high concentration, like here a saturated solution and a dilute peptide solution. You drip it on a metal target, you evaporate the solvent, and then you put it into the mass spectrometry. In fact, early on, we always got nicotine in the spectra because Michael was smoking in the laboratory, and he used his lighter to dry up the samples. So, you know, I mean, how do you in the early times? So, usually, it's not always so, but I have to be generalizing a little bit. It is a high difference between concentration, and it's in the end a solid solution. At that time, we already had changed to wavelengths of 337 nanometers in the UVA or 355, the tripled YAG laser wavelengths. And this was our solution to a good matrix. And actually, at the ASMS, I think it was 1990 or so in Miami, Brian Chiat, who was mentioned before, and who had set up a MALDI instrument, they were the first ones to confirm our results. And I were walking in the evening down the boardwalk and talking a little bit of what would be the next step to do. And we agreed that it would be advantageous to go from the far UV at 266 to UVA, and the reasoning was that we would most probably get less fragmentation because nothing would absorb there. In the end, it turned out, yeah, and we had no idea whether that would work. So we also told each other we had some matrices in mind, but we didn't tell each other what it was. And we decided we would both try to go for it. They were waiting for a frequency tripler, and we were waiting for a nitrogen laser at 337. And whoever, got his instrument first could try. Later during that summer, when the conference by Al Burlingame in San Francisco was approaching, I was getting nervous. And I asked because we hadn't gotten our laser yet, so I asked Ute Baar, who is the life companion of Michael, to set up the excimer pumped frequency doubled dye laser. At that time, this was something very difficult to do, but after 2 or 3 weeks, she got it to work. And we found out it works at that wavelength as well. We were lucky. The Rockefeller people were still waiting for the tripler. So we beat them by a couple of weeks. But it was a very friendly competition, you know. We intentionally did not reveal. But I have to say a few words about strange things about the matrix. One is that our first matrix was DHB, and their first matrix was very different. It's alpha-cyano 4-hydroxycinnamic acid. And even nowadays, I think 80% or so of the MALDI work is done with these two matrices, either of them. And hundreds, literally hundreds of different substances have been tried as alternative matrices. For some special applications, that's okay. But by and large, it is still the same matrices that we started out with, and I have no idea why that is so. Something else I wanted to mention is that, let me see, early on, actually, relatively early on, we had the impression that the proteins got embedded in the crystals of the matrix. Actually, this is a polycrystalline sample. It's a micro preparation, but it's always heterogeneous and polycrystalline. And so we had the idea it was incorporated. So Kerstin Struppert drew large crystals, millimeter-size crystals of the matrix, DHB, and then we cleaved it, and we got spectra, and we drilled holes into it, and we got spectra. So we assumed it was incorporated. At that time, at a meeting in Münster, I met someone who was considered the #1 wizard in organic crystallization, particularly bioorganic crystallization. In fact, he is a Nobel laureate for that. And I presented that to him, and I wanted to get his feedback. And his answer was, no way. This just cannot happen, and it will not happen. You have to be cleaner with your preparation, and. Well, I went out of that discussion going back, and we did a few more experiments, and then we settled for the idea that we were right and he was wrong, which is not, you know, when you're not that old and you have this giant in the field there, it's not so easy. By now it's clear it gets incorporated, and for the incorporated samples, it works better. You can also have it precipitate at the surface, but that's another story. Okay, so we also early on developed this very simplistic view of what the laser would do upon absorption. You know, there was this plume with matrix molecules and matrix ions and analyte ions, and the idea was that the ionization step was a proton transfer in the gas phase. That was the accepted hypothesis. And, but it's fair to say that compared to all the applications of MALDI, there was not all that much systematic work done into clarifying the mechanisms. And so it came later that we and others did at least a little bit of work. And let me quickly finish my talk by looking at that. But this was the first definitely non-volatile molecule, this melatonin, that you could not desorb without a matrix. And we took that in 85. So coming back to the question of mechanisms, let me see. Oh no, first, the other very surprising fact, and still surprising to me, is that the system seems to work on almost every type of molecule. This is a monoclonal antibody, it's a protein spectrum, this is a peptide digest, This is DNA spectra. You can only do smaller ones, but you can see in a TOF you can get good mass resolution for DNA 5-plex in that particular case. There are synthetic polymers, there are sugars, glycoconjugates in sugars, and there are small molecules. So the principle of MALDI, of matrix desorption, apparently is quite general. So let's get back for a moment to our simplistic view of the desorption event. Well, quite a bit later, we set up a very high-speed photography setup where we would look with nanosecond time resolution at the evolving plume, and that's one of the pictures we got. And if you look at the two, you would say, we were quite right originally, except that look at the time scale here. This was taken 5 microsecond, it was infrared MALDI actually, 5 microsecond after the desorption event. And if you look at the nanosecond scale, where the ions are generated that you see in the TOF, if they would keep generating ions for 5 microsecond after laser exposure, time of flight mass spectrometers would never work. So what looked like a proof of our original concept, it turned out to be really not true. And we still don't know exactly what happens in the desorption events during the first, let's say, 10 nanoseconds or so, which is typically the time scale at which the ions of interest are generated. Okay, about the ionization, gas phase ionization certainly appealed to me. It did, for various reasons, not appeal all that much to Michael Claras. And he developed a different model called the Lucky Survivor model, which is based on a pre-charge of the analytes in the crystal. And that's the only time in all of this development where a somewhat fierce and not friendly debate started about the ionization mechanism. And it was finally resolved about 2 years ago when one of Michael's PhD students derivatized alpha cyanomatrix by, how do you say that? He deuterated and esterified the matrix. So that way, and I don't want to go into it, it's Torsten Jaskola, he could differentiate between the method, and what's the result? Both are active. Depending on the laser fluence and the matrix and so on, one is correct and the other is correct, so there was had been no need for this fierce discussion. I would end up by saying the same that Peter was saying. Those years, actually most of the years of my professional careers were a lot of fun. There were times when we were the most happy group in town, and we would come in the morning with some ideas and try something and got a new result, and it was just fantastic. And to show how good we felt, I show you this here. This is at the Desorption Conference 98 in Brazil. And they had invited a professional samba group to perform. And at the end of their performance, they picked a few of us, and I'm sure you recognize this guy here, and this one, and we danced samba with them until at least I was totally exhausted. I was flat on the ground. These ladies have a very good physical condition. And for me now, it is time to pass the further development on to the next generation. And so I'll show you a picture where I'm waving goodbye to MALDI further development. It was on a parachute jump that my former students gave to me on my 17th birthday, and I'm enjoying it, as you can see. And as I said, I'm waving goodbye, but the further development is not over. I'm sure we'll see more of it and more interesting results. So thank you for your attention.
We would have time, say, for one question now. Remember, we can always have questions during the recess also. So, somebody want to ask a question now? Okay.
I'm always running over time. I know. I'm sorry for
You're
that.
perfect. Thank you, Franz. Okay. Thank you very much.